Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Acute Pain Models in Drug Development. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation and we'll try to attend to as many as possible. If you're joining us via GoToWebinar, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. And if you're viewing this presentation from our LinkedIn Live event, you can submit your questions using the comments tab on LinkedIn. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank Dr. Vince Clinical Research, who developed the content for this presentation. Dr. Vince Clinical Research, or DVCR, is a full-service CRO specializing in early-phase clinical trials for both healthy volunteers and patient populations. Their expertise spans neuroscience, substance abuse, pain, cardiometabolic disorders, infectious diseases, and more. DVCR offers comprehensive services including project and data management, biostatistics, medical writing, and site feasibility. Operating a cutting-edge clinical pharmacology unit with over 90 beds, DVCR ensures advanced capabilities and luxurious amenities to enhance participant recruitment and retention, delivering smarter, faster data to biopharmaceutical clients. What you're seeing on this slide here is one of 3 buildings on the Dr. Vince Clinical Research campus located in Overland Park, Kansas. And now, before we kick off the presentation, I would like to pass the mic over to Dr. Brad Vince himself to tell you more about DVCR. Dr. Vince, the floor is yours.
Great, thank you, Corey, and good afternoon, everyone. My name is Brad Vince, and I'm the CEO and Chief Medical Officer of Dr. Vince Clinical Research. I first want to thank everyone for joining this webinar today on acute pain models in drug development. I have the pleasure of introducing our 3 esteemed speakers for today's webinar. First, we're going to hear from Lynn Webster. Lynn is the Executive Vice President of Scientific Affairs at Dr. Vince Clinical Research. Dr. Webster is an internationally recognized clinical research expert in both pain and addiction medicine. He has participated as an investigator in hundreds of clinical trials and has authored more than 300 peer-reviewed articles. Dr. Webster is board certified in anesthesiology, pain medicine, and addiction medicine. Next, we're going to hear from Ira Gottlieb. He is the medical director and founder of Chesapeake Research Group. Dr. Gottlieb has over 25 years of experience as a board-certified podiatric surgeon and clinical researcher. He is the founding member of Chesapeake Research Group in Maryland. He has been the principal investigator and completed over 150 clinical trials. Dr. Gottlieb specializes in research focused on the relief of painful conditions and is also dedicated to finding new medications to help reduce the worldwide opioid epidemic. Last, we're going to hear from Rose Geringer. Rose is the ratings manager at Dr. Vince Clinical Research. Rose oversees subjective data collection at DVCR. She brings over 25 years of experience as an instructor in both corporate and academic settings to train staff and volunteers in the collection of primary endpoints for human abuse potential and acute pain models. Thank you again, everyone, for joining the webinar, and back to you, Corey.
Thank you, Dr. Vince. As you mentioned, our first speaker today is Dr. Lynn Webster, Executive Vice President in Scientific Affairs at DVCR. Dr. Webster, over to you.
Thank you, Corey. I'm going to address the most common human pain models. Next slide, please. Now, we have 3 objectives today. I'm going to discuss 3 of the major acute pain models, including the differences in the designs, the timelines, and some of the cost. The second objective is how the study designs for non-opioid versus opioid analgesics may differ, and there's only going to be a comment about that, one comment about it. And then most of our discussion today is going to be focused on the bunionectomy model, highlighting all of the key factors that are necessary to study and to determine success of the study. Next slide. As I said, I wanted to mention something about, really, the FDA has come out with some guidance on studying non-opioids and how to differentiate them from opioids in some of the acute pain models. You still have to demonstrate efficacy and safety if you are looking for drug approval, but there are additional considerations for non-opioid analgesics, and that as maybe primary endpoints, and that is the elimination of opioid use in the setting where opioids are usually prescribed as an objective of the design of a new non-opioid. Additionally, the adequate pain management that allows patients to be discharged from a hospital or somewhere without the need of opioids to be sent home with them. And then direct patient benefits from reduced opioid use, such as fewer opioid-related adverse reactions or a faster functional recovery, if that can be demonstrated. These are three hallmark endpoints, measurements that the FDA has indicated that they would like non-opioid analgesics to address sometimes, but you still have to demonstrate efficacy and safety, of course, to get approval. Next slide. A large number of pain models have been studied over the last 20 to 30 years. I've got the numbers here. Now, to get approval for an acute pain indication, the FDA has indicated that usually you need one hard tissue and one soft tissue model. The bunionectomy and the third molar extraction are hard tissue, and abdominoplasty is the soft tissue. There are other models, there are other soft tissue and hard models, but these are the three most common that are used. You can see that there have been quite a few of these over the last 20 to 30 years, and still quite a few are under use today. The active studies, you can see there are 6 currently as of the fall of 2024 and the first quarter of 2025, based upon looking at clinicaltrials.gov. And third molar extraction still over 30. And abdominoplasties, there are 12 studies undergoing. So, and there are others being planned, mostly for third molar extraction. And that's a little curious because it's not used generally for drug approval, that is a pivotal study. But there are real advantages to use the third molar extraction model, and I'm going to address that in just a moment. Next slide. And I think it's important to understand that all of these models have different strengths and weaknesses, and it really depends upon the drug that's being investigated, whether it's a non-opioid or it's an opioid-like or it's a mild analgesic, an NSAID. There is only one way to achieve a successful outcome, regardless of the model, and that is if the pain model is sufficient for an active drug to be demonstrated superior to a placebo group. So the bar that is set, if you will, has to be just right. So you have to choose the right pain model to produce the right pain stimulus in order for the particular molecule that you're studying to hit that sweet spot. If the active drug and the placebo exceed the analgesia, or I should say the pain intensity, they'll both fail. It's a failed study because they're going to be unable to be separated. Same is true as if the study design allows for too much pain so that even a potent analgesic will not separate from a placebo because it won't demonstrate enough efficacy. And we see that sometimes, even in bunionectomies, in studies where you're looking at the first 6 hours or so after an operation where the intensity of pain is the greatest. And that's why there are different designs for the bunionectomy, as you'll hear today. Next slide. So just as an overview, because you're going to get more of this from Dr. Gottlieb, or not this one, this is the third molar extraction. So just as an overview for the third molar extraction, because it's not our main focus today, I wanted to mention that this is generally an outpatient procedure, but when we when we use this model, the subjects are usually in-house for the period of time in which we're monitoring. One of the strengths to this model is rapid enrollment. It's the quickest among all of the acute pain models, and it's got very high assay sensitivity. It's been demonstrated over and over again to have very high sensitivity, but it has to be matched, as I just said from the previous slide, to the potency of the drug. You don't want a placebo to be able to demonstrate as much efficacy as the analgesic that is the study drug. The post-op pain experience can be adjusted with this, modifying the surgical protocols a little bit so that you can get more intense pain, and the way in which the drugs are being delivered, IV versus a placebo in multiple doses. But generally, the assessment period is under 24 hours because the pain lessens dramatically by 24 hours and beyond. So the interval of assessments for efficacy would be generally under 24 hours. It's considered a mild pain when you consider it comparable to or in comparison to some of the other pain models like a total knee or bunionectomy or abdominoplasty. But it is highly predictive of efficacy in later stage pain models, and that's why it's so commonly used. It's very useful in matching PK/PD. It's useful for onset of analgesia and to get a signal of its potential efficacy if the protocol is written well. Next slide. Now, the abdominoplasty is relatively new. If you remember on the earlier slide, there were no abdominoplasty models, at least that I could find in clinicaltrials.gov prior to some 20 years ago. There are different versions or varieties of abdominoplasties that can generate different levels of pain, and so you have to standardize your approach. But it is generally an inpatient, but it can be an outpatient procedure in the real world. But in clinical settings, we need to have, obviously, the patients in-house, domiciled during a period of time in which measurements can be occurred. The good thing is there's quick enrollment, but there's no disease or diagnosis required. Healthy participants generally, and a lot of participants seek this out because there are a lot of people who want to get rid of a little abdominal fat. Complications are low, but there are complications. And there's a strong post-surgical pain signal, certainly for 48 hours and maybe even 72 hours for potent analgesics. So it's a good model. Next slide. Now, we are going to focus on the bunionectomy, and I'll just be brief on this because Dr. Gottlieb's going to speak more about this. It is generally an elective outpatient procedure in the real world. In healthy participants, but for us, we domicile individuals, and the assay sensitivity is very good. It has a high assay sensitivity for up to 72 hours. So one of the differences here amongst the different models is what interval of time can we use to assess potential efficacy. This one has a longer period than certainly the third molar, and 72 hours is generally pretty good. And that you can generally recruit a large number of subjects in a limited number of sites. The good thing is that there are low rates of patient comorbidities associated with this. They're generally healthy people, as I say. It involves a cutting of the bone, an osteotomy. The protocols for the surgical as well as the anesthetic are standardized, which helps decrease variability and increases the reliability of the possibility of determining the real effect, the efficacy of a drug, and it permits different types of flexing and dosing, flexible dosing. Now I'm going to explain that in a minute. So the collection of the data has been standardized over the last 35 years. It's probably the most studied area, other than the third molar, for those are the two obviously most common pain models that are used. Next slide. Now, this is probably where most of the meat to my talk is that anyone interested in this webinar is going to find useful. And I'm looking at, again, the three most common acute pain models today. And I first break it down. I'm looking at enrollment rates, assay sensitivity, the common endpoints, timelines, model limitations, just a sample of it, of course, and the cost per participant, and then some of the other issues like advertising costs. Now, with the bunionectomy, there was a time when we could do probably 40 of these a month in my site, but it's becoming more difficult to enroll. So we're looking at now an average of maybe 20 participants enrolled per center per month. And so, you know, if this is going to be a proof of concept study or if it's going to be a pivotal phase 3 study, how many sites you might need or how long it will take in order to complete the study. Third molar is a much more rapid enrollment, so you can do over 60 participants, and usually fewer sites are needed because of the larger enrollment rates. And abdominoplasty is pretty good enrollment, better than bunionectomy, but not as good as the third molar. And again, you have to factor all of that in with the cost, the duration of time that you have to complete the study, and et cetera. Now, the effect size is remarkable for the third molar. You can see it's 1.51, and it's very good even for the bunionectomy. It's good for abdominoplasty. It's considered moderate response. So all three models are good. The third molar obviously stands out as exceptional. Common endpoints, it depends upon the design of the study and actually the drug itself. So, but I've listed some of these, like the TOPAR, which is the total pain relief, the number of responders, the use of rescue medication when it occurs. These are common across the board. The pain intensity difference, but you can only use a pain intensity difference. That would be the sum of pain intensity difference in patients or in a design where you will have a significant amount of pain at the onset. So it doesn't work if you, for example, dose preoperatively or intraoperatively. So your endpoints have to match what obviously the design is and the molecule and its intent and onset of action. Some of the limitations for a bunionectomy is that, you know, it's argued that sometimes it lacks clinical relevance to other acute pain models. It's a highly controlled environment, which is one of the strengths for sure. The safety and efficacy findings cannot be generalized to an elderly, hospitalized population or special populations usually. And the population is, as you will hear more about later, generally female. And then you've got the third molar. It's also not generalizable, and you know, pain intensity is less, so it's really more commonly used today for a proof of concept than it is for real-world efficacy studies. And it can be influenced a great deal by the local blocks that are used that can actually reduce the amount of pain intensity during a period when you want to measure or have a significant signal in order to show a difference. And then the abdominoplasty, this is a little slower recruitment, obviously, rates than with the third molar and maybe with some of the others. It usually requires several centers. And so there are limited sites that can do this. And there are some potential complications with this from the general anesthesia, but mostly from the operation itself, but limited. Now, what's the cost? Well, these are 2024 estimates that I've been able to come up with, and the range of, obviously there's a range, and it will be influenced a great deal by the protocol design, the molecule, the objectives, the endpoints, and whether it's a proof of concept or if it's a phase 3 pivotal study. There's additional costs like advertising costs and sometimes the completion of the CSR that have to be taken into consideration. Next slide. So the timelines for dosing paradigm depends upon when and what is being done. And this can apply actually to all 3 of the models used, and probably even some of the models that we're not discussing today. The top one is preoperative dosing. So you randomize the subject, then you give a dose, then you have surgery, and you measure the assessment over the next 40 hours. Now, there's no baseline pain to compare it to, so you can't use a spit. So it's going to be a different endpoint, and interoperative is the same way. You can randomize and then do the surgery in which you provide the study drug. And then you make the assessments over the next 48 hours, roughly, depending upon the model. Obviously, if it's a third molar, it would be just 24 hours. But if it's bunionectomy, it could be 48 hours up to 72 hours. And then the post-op dosing, which is the standard, most common, I should say, rather than standard approach. And this is when you wait until there's an adequate amount of pain. Intensity, and then you randomize them. So they may have to have a pain level, let's say, of 5 or 6, depending upon the protocol and what you're trying to do. And then you give your first dose, measure your assessment over time, maybe 48 hours, maybe 72 hours. And then there's another technique that's, I think, limited to only the bunionectomy. I'm not sure that it's used in total knee acute pain models, but this is when you have surgery, you place in a block, a regional block that we call a popliteal block that provides complete pain relief for about 24 hours. And then you discontinue the popliteal block, allow the pain to occur, and then wait until there is an adequate stimulus of pain reported. Then you randomize the subjects, they get their first dose, and you measure the efficacy over roughly 48 hours, whatever is necessary. And I mentioned that earlier, that it depends upon the model, what your objectives are, and as to whether or not you use any of these, and the drug. What is the pharmacology of the drug? What's the PK? What's the PD of the drug? So these are different considerations that you can have for all of the procedures, but the post-operative dosing is usually limited to the osteotomies, the bunionectomy or total knee, because you can put a regional block in. You can't do that as well with an abdominoplasty or with a third molar extraction. Next slide. So the timing of assessments, I've alluded to this already. If it's a pre or intraoperative dosing, you don't do that in the inclusion-exclusion criteria here. It's really post-operative dosing there. And the baseline assessment, there is none for pre and intraoperative dosing. Primary endpoints here are some pain intensity, but not a difference, not same, and that's the same thing as an AUC. So it's the amount of pain that someone's experiencing from whatever time you choose to begin until the end, and you do that in comparison to the placebo or an active comparator. But with the postoperative dosing, it is a sum of pain intensity difference, most commonly used. That is, wherever you start, that's the beginning pain, and so you're looking at a difference. So if somebody starts at a 5 or 6, and then it drops down to a 4, that's the difference that's measured over whatever period of time. AUC is also calculated with this model. So you can't really determine the speed of onset with a pre or intraoperative dosing, but you can with the postoperative dosing. That can be measured. Next slide. And then I think this is my final slide. I just wanted to give you a sense of the timeline estimates, and of course there are a lot of variables that go into this, and this is in no way fixed. but you've got to consider the protocol writing. Usually, I think of about 3 months. You've got IRB, and if it's a central IRB, usually you can do those in 2 weeks, sometimes less, sometimes more. But if it's a third molar extraction, those are rapid enrollment, as I have already alluded to, the data analysis, the CSR development. You can get that done in maybe 15, 16 months. The abdominoplasty is a little bit longer to do all of those, and you can see the minimum, maximum, or the range, and there's really no minimum or maximum here, but this is the general range. And the bunionectomy is a lot longer to complete. That's going to take over 2 years if you're at the beginning, protocol writing to completion, depending upon whether this is a proof of concept or if it's a pivotal phase 3 study and you're using 300 patients or 400 patients in 6 sites or something. It all depends upon a lot of variables, the inclusion, exclusion criteria. But this gives you a sense of a relative enrollment period and timeline with the 3 most common models that are used today to assess drug efficacy and safety for acute pain. Next slide. That's it for me today. Thank you very much. Back to you, Corey.
Well, thank you very much, Dr. Webster, for that really insightful presentation. We are now moving to our second presenter today. At this time, please welcome Dr. Ira J. Gottlieb. He is the Medical Director at the Chesapeake Research Group. Dr. Gottlieb, over to you.
Great, thank you so much. I appreciate the opportunity to speak with you all and discuss this topic. You can go to the next slide. So what we're going to talk about today is the bunionectomy model. And my goal today for today's discussion is to review the methodology of the bunionectomy pain model and to explain why it has been so critical in the FDA's approval of new, novel, and effective analgesics for the past many years. You can go to the next slide, please. So for the past 25 years, this model has been the preferred pain model used in clinical research studies, testing the efficacy of investigative products used to treat post-operative pain. The model has been used in studies investigating oral, topical, parenteral, and implantable medications randomized via many different routes of administration. So the bunionectomy is a very common disorder. It's present in diversely across the population between 18 and 75. And the surgical procedure, go to the next slide for a second, please. The surgical procedure is performed in an outpatient setting. So the outpatient setting involves an ASC. This using the ambulatory surgery center and our clinical trial site as the location for the study allows us to use central IRBs rather than institutional IRBs. The bunionectomy surgery itself is a relatively short procedure lasting between 30 and 60 minutes. The surgical procedure has been standardized to minimize variability between surgeons and research centers around the country. The procedure requires IV sedation with the infiltration of local anesthetics with or without continuous nerve blocks. There is no need for general anesthesia or intubation with this procedure. The evolution of and the results utilizing the bunionectomy model have been replicable and reliable over the past 25 years. Next slide, please. The bunionectomy model has been the preferred pain model used in clinical research studies evaluating the efficacy of investigative products used to treat postoperative pain. Most of the recent FDA medications approved utilized the bunionectomy model for their pivotal trials. The bunionectomy model itself is remarkably flexible. It has been used in studies testing products with different routes of administration, including oral, topical, parenteral, injectable, and implantable medications. It has been used to test medications expected to relieve moderate and/or moderate to severe pain. The model's flexibility allows for randomization, hence administration of the study drug. We can randomize subjects with this model either prior to surgery, during surgery, day 1 after completion of the surgical procedure, and on day 2. These day 2 studies typically require the use of continuous regional anesthesia with nerve blocks. Further, the NRS pain score can be selected based on the sponsor's preference for randomization. It can vary between 4, 5, or 6 out of 10. The pain score is always in the moderate range. You can go to the next slide. So before I get to the procedure itself, I just want to mention that there are other bony models out there, and these are called large joint models. Again, large joint models. And I'll give you a quick overview of some of their characteristics. So examples would be the total knee replacement, total hip replacement, total shoulder replacement, and spinal fusion procedures. These models typically rely on an older demographic with greater comorbidities. They require longer to recruit and thus longer duration for study completion. These studies also are much more expensive just based on the invasiveness and the cost of implants, and they do not have quite the sensitivity, consistency, or reliability compared to the bunionectomy model. It also requires longer surgical times to complete the procedures, and there is a greater incidence of adverse events and serious adverse events, such as DVTs, pulmonary embolus, CVAs, MIs, and complications related to the procedure. Back to the bunionectomy model and its flexibility. Over the years, the early bunionectomy trial involved randomization on the same day as surgery. Experience taught us that in some cases, the pain signal on day 1 was too strong for some investigational products. Depending upon the investigative product, we can now select randomization on day 1 or day 2. As mentioned earlier, the day 2 randomization requires typically regional anesthesia with continuous nerve blocks. Specifically, the overnight study, the day 2 model, requires the block to be discontinued between 3 and 4 a.m. the next day. Subjects will then develop over time moderate to severe pain as the block wears off, which would then trigger the selected NRS score for randomization. Typically, randomization must occur before 9 hours after the discontinuation of the block, and the in-unit observation period varies between 1 to 3 nights and 2 to 4 days. So let's dive into the surgical procedure itself. And why does this procedure consistently trigger moderate to severe postoperative pain? First of all, the bunionectomy surgery requires manipulation of many different tissue types. There is a skin incision placed dorsally or medially over the joint. Subcutaneous tissue is dissected, and retraction of neurovascular and tendinous structures is necessary. The joint capsule and the highly innervated periosteal layer is incised and reflected. The medial portion of the enlarged metatarsal head is removed. An osteotomy or bone cut or fracture is required to reposition and reduce the first intermetatarsal angle. The osteotomy is then stabilized with internal fixation, typically using screws. Lastly, there is a 3-layered closure to complete the procedure. Next slide, please. Great. So these are x-rays demonstrating the pre, immediate post-op, and post-op x-rays at 30 days. So these x-rays demonstrate the correction of the angulation of the great toe, as seen on x-rays 2 and 3, as well as the angulation of the metatarsal and the placement of the internal fixation. The need to dissect and retract and manipulate these many structures results in the moderate to severe pain, which plateaus typically for about 72 hours. Next slide, please. Let's talk for a few minutes about the recovery. Initially, after surgery and for the first 4 weeks, subjects are required to protect the surgical site in a partial weight-bearing orthopedic boot. After this time, we generally obtain x-rays to ensure satisfactory alignment. And from weeks 4 through 6, we will provide a surgical shoe to allow the patient to ambulate as they then soon transfer into a walking or running shoe. This usually occurs at about 6 weeks, and the subjects may return to their regular level of activity, weight bearing, exercising, etc., as tolerated. Next slide. Again, I thank you so much for the opportunity to discuss this pain model. It has really been I guess you would say an anchor in the development of these new pain relieving medications. Thank you very much.
Well, excellent. Thank you very much, Dr. Gottlieb. And now please welcome our final speaker for today. This is Rose Geringer. She's ratings manager at Dr. Vince Clinical Research. Rose, over to you.
Thanks, Corey. I'd like to turn our attention now to some of the challenges as we screen, assess, and train volunteers to take part in these great clinical trials. The next slide shows that we've got subject considerations and environmental consideration. There could be challenges when we're talking about inviting people to come and participate in clinical research. They're bringing all their past experiences with them. They're going to all have different experiences with pain. Some suffer from chronic conditions, others have had serious injuries, which gave them experience with acute pain. Their individual tolerances are going to be based on those experiences and their neural receptor health. Of course, we all have expectations of ourselves. Some of us try to tough it out and be macho. Others are fearful and anticipate a lot of unmanaged pain. And so we want to take all of that into consideration as folks come in to help us with these great trials. We had one gentleman, for example, he had some peripheral neuropathy going on, and he had already had some significant injuries in his past. And so because of those, he didn't actually qualify for a pain tolerance because he was unable to feel extreme pain. When we think about environmental considerations, we want to standardize as much as possible. We have all of these subject considerations that they're bringing in. So we want to have an environment where we can standardize each one of those processes and get great data for the companies that we work for with these new pain medications that are so important for our communities. So we standardize the procedure by limiting the number of staff interacting with the patients or volunteers. We have a private recovery setting so that they are not influenced by other people that might be having the same surgeries. And we want consistent administration. For example, before each pain assessment or other assessment they might be doing, we're going to give the very same instructions every time. It just makes it more scientific. On the next slide, you can see that we have some recruitment considerations. When we have healthy, normal volunteer studies to just study the efficacy or safety of a regular medication, we have very narrow recruitment parameters. For the bunionectomy studies, we've widened those out a bit because the incidence of bunions is in an older population. And so you can see listed here, we have various recruiting benchmarks for these volunteers. We need to check and see if they have good venous access. A higher BMI might include some other preconditions that might be a factor, like that gentleman with the neuropathy issue. We also have found scheduling conflicts to be a difficulty in recruiting these studies because, again, as they've said, it might be 6 to 8 weeks before they're back to their normal schedules. So in addition to scheduling the surgery itself, they have to have that room in their lives in order to have a good recovery. So those are some of the common reasons for failures as we screen folks over the phone. In addition to our regular database of recruiting subjects, we have reached out to various areas of the community to find people that might qualify for this surgery. We know that 85% of these folks are going to be women statistically, and so we want to reach out to men as well. And so to get a better demographic, we have gone out to different modes of advertising, not just our standard electronic TV and radio ads. We've also used billboards, banner ads on email platforms, and even Craigslist in order to recruit these subjects. On the next slide, we have some key considerations for those inclusion criteria. So again, typically in clinical research, we keep a very narrow age range. For the bunionectomy studies, we wanted to expand that to include more people. We also have the inclusion criteria that Dr. Gottlieb mentioned, where if they have that nerve block in, we remove that overnight, and then they have 9 hours to qualify. I mentioned the gentleman with the peripheral neuropathy, and that he did not qualify in those 9 hours. So it's a key inclusion criteria. They have to have a certain amount of pain in order to really effectively rate the pain management that is received by the new medication. So when we exclude subjects, there's a list of the exclusion criteria. And of course, anybody who's had surgery in the last month or is taking other medications, they cannot be part of the study. We want to have good, clean data at the end of the day. So you can read through some of the other exclusion criteria that we implement for various protocols. Next slide, please. Using the bunionectomy model, we're trying to find effective and safe medicines. And so we have assessments that determine how effective those medications are. We have clinician-rated assessments, including the modified observer's assessment of alertness and sedation. So we're checking to see how affected is the patient in their sedation. Is it hard to wake them up? Are they really out of it? The Columbia Suicide Severity Rating Scale is again done by a trained clinician to determine if there is any differences in any suicidal ideation because of the medication. We might also use a global impression of change. The clinician has seen them from the beginning to the end of the study to assess their overall performance. And then we've occasionally had assessment for the staff satisfaction with dosing. Sometimes it's an intravenous dose, and that can get a little bit more complicated than, say, an oral dose of medication. In addition to having those clinician-rated assessments, we also have those participant-rated assessments, the numerical rating scores. This is how we know how effective the medication is overall. So sometimes the assessment wants them to say, at this moment, how much pain relief are you receiving, or how much pain are you experiencing? And it's always at the surgical site. We have to remind them in those standardized prompts that even though they may have been laying in bed for a few hours or maybe 48 hours, and that can get a person a little stiff and sore, we want them to only rate the pain that they're having at their surgical site. We can use stopwatches to determine how quickly the pain relief is felt. So we can have a stopwatch that they will stop when they have perceptible pain relief, and then another stopwatch that they can use when meaningful pain relief is achieved. A global impression of change we can use, just like the clinician, the patient can also be asked how they felt their recovery is going. We might ask them to rate their pain at rest or pain with movement, and if there's any nausea or vomiting that they might have experienced over time. The numerical rating scores and the visual analog scales have been validated to use in studies to find out exactly how much pain a person is experiencing. Those are both great assessments and really the key criteria for determining the efficacy of the new drug. On the next slide, we can see some ways that we can reduce the variability. We have different staff that are providing different roles in the study. We have caregivers who are doing everything from assisting the volunteer to use the restroom, getting their meals, any personal hygiene items that they might need. They're keeping them comfortable and happy in the clinical setting. Our raters, on the other hand, are those people who have very limited interactions because we don't want them, our demeanor to affect their rating scores. So raters should have minimal interaction with the volunteers. We want our demeanor to be professional, but yet impersonal. We don't want to dote on them and make them feel, sweet and happy because they're there to provide good clinical data. But our caregiver roles are, of course, going to make sure they have everything they need and they are kept comfortable. The raters are going to use a standard prompt before each rating. We're going to ask them if there's anything they need before we leave the room, because they must request additional pain medication. It is not going to be offered. When people go into the hospital and they rate an 8 or a 9 or a 10 to the attending nurse, that nurse is on her way to the pharmacy, but that is not the case here in clinical research. So the environment, we want to reduce variability by giving them a private room so that there is no crosstalk among patients about how their recovery is going. And 15 minutes before each assessment, the rater comes into the room, gives them that 15-minute warning, asks them to set their phone aside so that they aren't influenced by any text or calls from friends or family that might encourage them to either request more pain medication or discourage them from that as well. Our subjects need to comply with the resting for 15 minutes. We don't want them up and about because, of course, that's going to affect their pain score, and we're only going to be rating the pain at that surgical site. Next slide, please. So we want to have accurate assessments so that we can determine if these new formulations are going to be effective for pain relief. So we want to train the subjects on what to expect. Of course, they've gone through an extensive informed consent process, but we want to familiarize them with those numerical rating scores and also other assessments that will be happening during the course of their stay at the clinic. We're going to train them before surgery, immediately after surgery, prior to that first dose of what assessments they'll be completing and what's expected of them. Often they go home with a diary, either electronic or on paper, so that they can continue to complete those pain recordings for rating the effectiveness of the medication. Subjects are trained to only rate the pain at the surgical site, not to discuss with family or friends what is going on at the clinic or if they should be getting more or less pain medication. And we encourage that by having them set that phone aside for 15 minutes. And during the ratings themselves, we could eliminate all the distractions, no staff moving around the room. It's just the rater and the volunteer making those assessments. Again, we include when we leave the room, that if there's anything they need, they need to let us know. So with that, I like the final slide here. I think it's time for some question and answers, perhaps. Corey, back to you.
Well, fantastic. Thank you, Rose, and thank you all very much for that insightful presentation. We are now ready to begin the Q&A portion of the event, and as we do so, I invite the audience to continue sending their questions or comments. Just a reminder, if you're attending via GoToWebinar, please use the questions chat box, and for those of you watching on LinkedIn Live events, feel free to post your questions using the comments tab on LinkedIn. It looks like we have already received some audience questions, so I'll go ahead and kick off with the first one, and I think this one's probably best sent to Dr. Gottlieb. Dr. Gottlieb, this audience member was asking if there are any patient characteristics that would cause less or more pain following a bunionectomy that should be considered.
I think using the experience and talking with patients prior to surgery and just interviewing them with what their experience has been with prior procedures, how they managed after the procedure. Did they find that their pain was severe, moderate, longer acting, shorter acting, and just getting a sense of their personality. I think that's really the greatest, it's the greatest sign or the greatest information to provide us with, will this patient have more difficulty after surgery than average?
Thank you so much for that answer, Dr. Gottlieb. Looking at some other questions that have come in here from the audience. This next question I think is probably best suited for Dr. Webster. So Dr. Webster, I'm going to pass this one your way. This audience member is asking about what are the potential errors in conducting the bunionectomy study?
Oh, gee, there are numerous ones. That's why we need to, yeah, I think the sponsors need to be sure that they've chosen sites that have a lot of experience, because almost every step, you know, protocol writing, I've recently seen a protocol that was poorly written, and it was loaded with potential errors because of that. But I think the most common operational concerns we have is for the subjects to understand the difference between pain intensity, reporting their pain intensity, and the pain relief. For those of us in the field, of course, we know what the hell that means, but it's not clear to subjects, what do you mean pain relief? You mean now, or you mean from when I had the worst pain, or when I walked into the building? And that all depends upon the model, right? If you're giving intraoperative or preoperative dosing. So having clarity about that is important, and making sure, as Rose explained, the education of the subjects is really critical, so that they understand, all of them understand the same thing, to decrease the variability. Then I've also observed and experienced having some really wonderful nurses who care a lot about their patients, or I should say, they like caring for their patients, and these are subjects. So there's a difference between a nurse caring for a patient and a nurse observing a clinical trial. And the difference there, the big difference, is the empathy, because we know empathy can produce a significant placebo effect. So we need to be sure that our staff, the ones who are doing the assessment, the nurses and et cetera, are not expressing any real concern or empathy towards the subjects, not being rude or anything, but you have to have a neutral presence, a neutral appearance to them, in order to not influence the measurements. And then I think another error is that sometimes there's a lot going on, and the staff will not collect the data exactly when it needs to be. And so there is sometimes a tendency to go backfill, which is a big no-no. And so you've got to be careful that the data collection is exactly within the window that has been in the protocol in which it's to be collected.
Great, thank you for that answer, Dr. Webster. I think we have time for a few more questions here from the audience. Just a reminder though, if we don't have time to attend to your questions, the team at DVCR will be able to get in touch with you after the webinar. This next question that's come in, I'm going to keep things with you, Dr. Webster, for this one actually. This audience member is asking about what are some other acute pain models that are used and why would they be considered?
Well, I just mentioned that I did mention during my presentation that the total knee arthroplasty is one that is used, and the reason for that is because it's a real world. You know, it's less standardized, which makes it a disadvantage to a large degree because there's a lot of variability, a lot of comorbidity with the population. Total hip arthroplasty is another one. These are two hard tissue models. And for soft tissue models, there historically hysterectomy, but there's less pain generated with hysterectomies, and I haven't seen a hysterectomy study, a soft tissue study, in a long time. But there are herniorrhaphy models and hemorrhoidectomy, and so why those would be used would largely depend upon the drug that's being studied, because they may want, for example, a local anesthetic as opposed to an oral agent. And the local anesthetics make themselves more amenable to being placed in and around the wound of hemorrhoidectomy or herniorrhaphy. So we're always trying to match the protocol design with the pain model and the pharmacology of the drug. And those are the reasons.
Thank you so much for that answer, Dr. Webster. Moving along here through the Q&A, this next question that's we've had come in, I think is probably best suited for Rose, so I'm going to send this your way. Rose, this audience member is asking about how you ensure diversity in recruiting for the bunion model when females represent most of the population qualifying for a bunionectomy.
That's a great question. And we do want to have as diverse a population as we can to come in and try these new pain formulations. And so we've really expanded our typical advertising out into the community. We've gone as far as putting up flyers in nail salons and theater programs, and we've really targeted certain social media areas with the geofencing so that we're reaching out to a diverse population, hoping to, again, get those very small percentage of men in the community that do qualify for the bunion surgeries to come in and participate in clinical research.
Fantastic, thank you, Rose. Unfortunately, I think this is going to be the last question we have time for here today as part of the webinar, but rest assured, if you've sent in your questions, the team at Dr. Vince will have a chance to follow up with you after today's webinar. This last question here, Rose, I think I'm going to send this one again your way. This audience member was asking about how do you control these subjects influencing other subject report of AEs and report of pain?
Another great question. Adverse events and the influencing of other pain scores is something that would be very common if it was a group of women sitting around in a room. And that is why we give each of them a private suite here at DVCR, so that really they are almost sequestered in those suites. We're minimizing their ambulation. They have a private bathroom to use. And so they don't interact after they arrive in our lobby and maybe a little bit of the pre-op. Each of those volunteers is going to be in their private room, just interacting with a few staff members.
Thank you very much for that answer, Rose, and thank you to all of our speakers for your answers during our Q&A session here. Unfortunately, we have reached the end of the Q&A. As I mentioned earlier, though, if we couldn't attend to your questions, we will have a chance to follow up with you after the webinar. But if you have any further questions, or if you're someone who's watching the recording of this webinar and you'd like to get in touch, you can reach out to the team at DVCR, at Dr. Vince Clinical Research, using the email address that's currently up there on screen. Very simple, info@drvince.com, and they'll have a chance to follow up with you. I do thank everyone once again for participating today. If you're attending via GoToWebinar, I've added a link in the chat box where you'll be able to view the recording of this event, and you can of course share this link with your colleagues so that they may register to view it as well. You'll also be receiving a follow-up email from X-Talks, and after we wrap things up here, a survey window will appear on your screen. Your participation in the survey is always appreciated as it helps us to improve our webinars. Just a note for those of you watching on LinkedIn Live, we encourage you to create an account on xtalks.com and register for this event, which will then not only give you access to today's recording, but to our entire library of content, including other webinars, articles, interviews, podcasts, and so much more. Now, I'd just like to thank the speakers today for their excellent presentation. Thank you to Dr. Vince Clinical Research, DVCR, for partnering with us on this. We hope our audience found it informative, and we wish you all a great rest of your day. Take care, everyone. Thank you.